And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Don't tell me.
We are live.
Are we live right now? Okay, well, it looks like we're live then. Awesome, guys. We got a good stream here tonight. We're gonna try as much as we can to kind of like our brother in Christ, Dr. Ken Hovind does. He does his Whack an Atheist streams and videos once a week. We're going to try and do it more frequently. It's just going to be certain videos that people send us that they feel need refuting. I've already got a long list of videos that people want us to address. A lot of the videos are just stuff that we've covered in the past, and that's fine because there's a lot of videos out there promoting evolutionism and, you know, attempting to refute creation, young earth creation, for example. So in due time, we're going to try and get to as many of those videos as possible today, I should say tonight, a little bit late, but we've got a good video by Biologos, what they believe is the best genetic evidence for evolution, universal common descent. So we're going to, a lot of this is going to be off the cuff. For example, we're just going to go through it. I think probably like you, Jeremy, I've skimmed through it a bit. I haven't watched it in a while, but we're going to try and take it line by line, point by point, kind of just have some fun with it. Maybe Praise can play a couple minutes at a time, 2-3 minutes at a time. We'll give our thoughts, kind of like we did with our Godless Engineer video last week. So I want to say to the audience, if anybody has a video that they feel like needs to be addressed, send to myself or anybody on this team, and we'll kind of add it to the list as videos we want to refute. Also, I want to give a shout out to the debaters yesterday, Salvatore and Ron, for a really, really good debate discussion. Lots of good feedback. We're hoping to have Brother Sal back on within the next week or two for another lecture. So that was a really good debate. And announcement-wise, it looks like we just confirmed it. John Maddox and myself will be debating on Modern Day Debate this Sunday. It'll be a 2 versus 2 against Fight the Flat Earth, I think is the one atheist name. And the other one I think is called Mr. Skeptic or something like that. You can check MDD for the advertisement. So that's something to look forward to as well. So anyways, yeah, I've got some good brothers in Christ here with me. We got Praise, of course, Jungle Jargon, Gavin, and Jeremy. Thanks for joining us tonight, guys.
My pleasure, brother.
Okay.
Welcome, Mr.
Hey, brother Gavin, good to see you. So, anyways, why don't we get this? Why don't we get this party started?
Here we go.
Like I said, so we'll have Praise play the video a few minutes at a time. Jeremy, you give your thoughts. We'll give our thoughts, and we'll get through as much of this as possible.
Yeah, just jump on if you want to say something. Here we go.
Chromosome.
Yeah, chromosomes. So the audience can see us, right? At the left
As
side.
discussed in our last
Okay.
Biologos podcast, the fossil record, while not entirely complete, reveals exactly the patterns we'd expect to see if Darwin's theory of evolution were correct. As more and more fossils are discovered, the evidence points toward the same conclusion. Species evolved gradually over billions of years, beginning with a single common ancestor. This conclusion, well supported by the fossil record, is supported by a vast amount of non-fossil evidence as well. One significant source of evidence is the field of genetics. To put it simply, genetics is the study of heredity and variation in organisms. Scientists that study genetics are concerned mainly with DNA, the molecule pretty well known by most people, that gets passed down from generation to generation. DNA determines which proteins are made in any given organism, and an organism's complete set of DNA, called its genome, is what makes it unique from all other living things. In the past few decades, scientists have uncovered an enormous amount of really useful information by studying genetics.
I mean, did they just admit? Okay, they just said that every organism's genome makes it unique.
Unique.
Didn't
Yeah.
they just admit almost something that contradicts their
Right.
theory?
Yeah.
It's kind of like when they'll call a biological structure design, or this whatever XYZ is designed for this. So it's like they don't even realize that they themselves is admitting a position that they don't hold to, right? So yeah, they're saying that each organism's genome is unique, but as she kind of iterated in the beginning, every single organism today ultimately descended from a single common ancestor billions of years ago, right? So I always point out that statement where they take what they observe today and conclude that all life is related is an extrapolation fallacy, right? So yeah, that's a good observation, Praise.
Yeah, they just seem to contradict themselves all the time. Go ahead, Jeremy.
Yeah, I saw pictures of transitional forms. Do you think we should say something about that?
Yeah. You know what? Whatever comes to anybody's mind regarding the video and what's being said, you're free to make your point. Yeah. What did you want to say about that, brother Jeremy?
Well, I just woke up. Okay. All right. I'll make it quick.
Yeah.
For example, on the whale evolution, as we know, each whale in their evolutionary artwork consists of only few fragments of bones, only assembled to look like whale evolution.
Right.
We are all familiar with this, and don't forget. Again, I want to repeat because most young YE creationists are forgetting that all of it, 100% of all the supposed transitional form of all animals found only have less than 40% of the bones found. And Lucy, their best example, is even less. It has most of the bones, and some even say it's not 40%, but more or less it's only 40%. Okay, and many of those bones are also in dispute, and I mean per bone. Okay. Also, is the audio doing well?
Yeah. And they're
Okay,
not even the
sorry.
same. They're not even from the same animal, the Lucy, isn't it?
Right.
Yeah, yeah.
Well, especially
Also,
because a lot of those bones in the the so-called ape to man ancestry, that series there, a lot of them, like if you look at the so-called Australopithecus sediba or Homo habilis, they are just ghost taxa. They're literally just a waste basket of random bones, some Homo bones, some ape bones. It's like, hey, what do we do with this? We don't know what this belongs to, so throw it in the basket and we'll call it Homo habilis. So much of the fossil record and so much of the interpretations are just based on, like you said, it's so fragmentary that they can pretty much, tell any story that they want to. Go ahead, Jeremy. Go
yes,
ahead.
yes, 40% of the bones only. And I've said before, it will never pass in court. I don't know if Mr. Hovind said it. I think he did. If it's presented in court as evidence, it will never pass. Okay. Now
Right.
I'll give a small example. Just one example that came to mind. Do you guys? All right. I'll not ask questions, so that this will be quick. Pak, Pakicetids. Pak, Pakicetids. Actually, it's an extinct whale. It
Pakicetus.
consists.
Pakicetus?
No, no, no. Pakicet. Pakice
Right.
Tids.
Pakicetus. Yeah.
Sorry. I don't know how to pronounce it in English. It's
Yeah,
an extinct. It's actually an extinct whale, as you know, standing for truth. It
Right.
consists of major it consists of a major modern whale body parts, actually, when they studied it. Okay,
Well,
so it's
Pakicetus. Well, I'm not sure if you're Pakicetus. They once
okay.
illustrated as like a full-blown cetacean whale, for example, but it's undergone such revision since then, where now it's just illustrated and
It's
promoted as
extinct.
just like a small, almost wolf-like creature. So yeah, there's almost nothing about Pakicetus that is transitional in the cetacean evolutionary stories. Is that the one you're referring to?
Uh, e- I,
Because
I'm not sure because
well, you've got
what
the
is the
Pakicetus, Ambulocetus, Basilosaurus, Rhodocetus, but Pakicetus they've got I think right at the beginning of their series of whale evolution there, and it's just illustrated as really small wolf-like creature that they used to promote as like fully aquatic because like you said they're only looking at these bone fragments, and looking at a bunch of bone fragments the evolutionists can tell any story they want. So once they discovered more bones for the Pakicetus, they realized that there's nothing aquatic about this, so it's undergone a really drastic revision.
okay. So it's Pakicids. That's the spelling, correct spelling, right? Or
Yeah,
I'm not sure if we are.
they'll say that the modern cetacean lineage has passed through something like a Pakicetid. I think that's like the group of animals that they would refer to the Pakicetus as. And the Paki goes from the Pakicetus up through the Ambulocetus to the Basilosaurus, all those different steps. But the Pakicetus is what's, and for the audience sake, I mean, you can just look up Pakicetus and you're going to see what they illustrated as. It just looks like a little wolf-like creature. You can see diagrams where it's the Pakicetus right next to something like a blue whale, and it just looks ridiculous.
It looks like a whale.
All right, guys, let's get going here. I just had to do it first minute. Here we go.
They've determined the structure
Yeah.
of DNA, how it's packaged, and how expression of it is regulated. Incredibly, they've even sequenced the complete genomes of hundreds of species, including humans and chimpanzees. All this information about DNA has revolutionized how we think about science, medicine, and even psychology. It's also changed how we think about common descent, the theory that all living organisms on Earth, from dandelions to mountain lions, descended from a single common ancestor. Genes, the pieces of DNA that are read by enzymes and translated into proteins, are the material upon which evolution works. Basically, this is what happens. Genes every once in a while accidentally become mutated. Because they are heritable, or passed down from generation to generation, a mutated gene can slowly spread throughout a population as time passes and as organisms reproduce. Harmful mutations eventually disappear because they make an organism less likely to survive and reproduce. But beneficial ones have the opposite effect. If all goes well, they spread until every member of the population has them. The result is a change in the population's characteristic set of genes. This change is called evolution. In Darwin's time, the theory of common descent was best supported by applying logic to what can be seen in nature. But now, we can also study something that's a little harder to see, DNA, the very material that makes living things develop in specific ways. Studying DNA has revealed an enormous amount of information that supports the theory
I
of
mean, does
common
anyone catch the equivocation fallacies? I mean, no one doubts, not even creationists doubt that there's change on a limited scale. And we would say these are neutral. These are on a horizontal type of creation. I mean, change, but
That's the only thing they can point to. They can only point to changes from what's already there. They can't account for how the information got there in the first place.
It's funny because, and praise, anytime someone else is talking, I just recommend you mute yourself, because if you're not muted, then there's that echo.
right.
But yeah, it's funny because you can see it there too, that, they just imagine, right, quote unquote, that mutations make something new and better. They'll just throw it out there like it's evidence for fisherman evolution, right? Praise, are you muted, brother? Are you muted, Praise?
Yeah, I just muted myself for the last minute or so. Yeah.
Oh, because I can hear that echo, but it could be coming from, I would just recommend whoever's not talking, just everybody make sure they're muted so we don't have that echo. But I think so the evolutionists, they're going to look at all these small scale variations, and then that's where they commit their extrapolation fallacy, because they think all of those mutations that accumulate that are neutral, for example, they're eventually going to add up enough to make something new and better. That's why they need lots of time, right? To them, time is the hero of the story. But yeah, that's a good point. That's a false equivalence phrase, because we don't disagree with changes in allele frequencies and populations, for example. But what's funny is that change is opposite of what is required to take that, single-celled ancestor into something like a whale over billions of years. So great observation there, Praise.
Yeah, I mean, they're the ones that are inputting their assumptions into their belief system. We're actually the ones who are actually practicing science that's observable, testable, and repeatable. That's what we see. We're not inputting all these presuppositions into that.
Right.
Descent. And amazingly, this evidence confirms what Darwin concluded simply from observing the macroscopic elements of nature, like the beaks of the famous Galapagos finches. To fully explain,
I mean, it's a freaking bird! Who cares about the stupid beak?
It's just general adaptation that they look to. And then it's like in the textbooks, right? They'll show a bunch of examples of microevolution, and then at the end of it, they're now concluding and telling the poor kids, hey, see, this is why you're related to banana plants. But we're not just looking for general adaptation or just random changes in allele frequencies, for example. Like, we're looking for how it's possible to explain the origin of, like, literally new forms of life from more simple pre-existing forms. Because if you're going to go from your single-celled like ancestor to a fish to an amphibian, reptile, mammal, bird, monkey, man, like, that's going to require a lot of new, novel, functional information, right? The evolutionists like to harp on the word functional, or on information. But if it's that which is specified and functional, any change of these mutations that they speak of on an area in the genome that's functional, that's important, like, that's going to be negative. That's going to be deleterious to the organism. So you're not going to take your fish to fisherman that way. Like Jeremy was talking about the whales earlier. Just think about how many incredibly complex adaptations would actually need to arise to change that small like land mammal, the Pakicetus, into a fully aquatic whale. And Praise, we've gone over it numerous times. I mean, you're looking at the emergence of a blowhole. You're looking at, I mean, even just the eye itself has to be modified for underwater vision. A huge one is the change in the birthing process.
Blubber,
where now the fetus is actually delivered in a breached position, and now the whale has to nurse its young underwater. I mean, we can go on and on. The origin of tail flukes. So these mutations that we know are generally deleterious, they're saying are the so-called created creative force. This undirected process is going to produce all these complex adaptations. Yeah, no, that's the extrapolation fallacy, and that's the philosophy when they take the small microevolutionary variations we see today and extrapolate that to say, hey, listen, over deep time, this tiny little wolf-like creature, Pakicetus, evolved into something like a blue whale. Like, that's the philosophy. So good point, Praise.
Yeah, I see these as morphological changes, not anatomical changes. See, the whole anatomy of the creature is not changed. It's just the morphology of it. That needs to be made known, too.
Good point. Good point.
Genetics supports the theory of common descent so convincingly it would take weeks on end. There's a lot of evidence here. Instead of trying to cover everything, today we'll just go over a few especially interesting points. Species thought to share a recent common ancestor are expected to have genomes that are extremely similar. Chimpanzees and humans, for example, are believed to have branched off from a single population of ape-like beings about 6 million years ago. Now, when we're talking about evolution, 6 million years is actually a really short amount of time. So if this theory is true, the genomes of chimpanzees and humans should have a lot in common. But one major characteristic of human and chimpanzee DNA is strikingly different.
I wanted to bring this up real quick. Raw Matt has done several streams and series on this, on the differences between apes and humans. I recommend people check that out.
Yeah, there is a lot of difference, and the way you're going to find out is to compare the genomes, and that's the only way you're going to find out what's a different kind, is because it has to be a copy if it's the same kind, or the same information. And when you compare chimps and humans, they're not copies of the same information.
That's a good point.
I'd just like to add a.
Oh, go ahead, brother Jeremy.
Yeah, don't forget that the genome are not sequenced entirely due to budget problems. So their evolutionary preposition again played a huge role, as they assume human chimp and ancestry, and human chimp came from gorilla, and then 6 million years elapsed right after that. So they branched off from a gorilla. Now I recall a quote from an evolutionist in 2017 or 2018. This is not a direct quote because I forgot the direct quote. It's in my archives again. I cannot find it. It says something like this. Okay. They say they are hindered by. Is there a problem with the volume?
Yeah, there is. So anytime somebody's not muted while another person's talking, there'll be so Praise, for example, is not muted, so that's why there's an echo. Just Praise. The echo comes from your side, brother.
I think I know what the problem is. Whenever Praise is on, has another channel open, that's when the
So
echo.
do I have to repeat again?
Are you on another channel, Praise?
On StreamYard I am, but not.
So do I have to repeat again what I said?
I don't want out Praise.
So Jungle Jargon, are you saying anytime Praise on another channel?
Yeah, I'm off mute. I'm off mute now. But yeah, I'm on. I'm on my own StreamYard right now, but I'm not even live. It's just it's a dead stream. So
do I have to repeat what I said?
It definitely comes from your end, Praise. Just watching our videos just to try and figure out the problem. It's definitely coming from your end.
I think it's StreamYard itself.
There is really echo, a huge echo.
it's still echoing even with me on mute, though. That's the problem.
So
that's what I think. It's the StreamYard system or whatever. Somehow it echoes through the system.
Looks like it's fixed now, Jeremy. You wanted to finish your point.
So I'll repeat what I said because the echo I think made the audience not understand what I was talking about.
Yeah, the echo is definitely distracting, so that's good.
I didn't mean to out you.
My echo. No more echo. No more echo.
Nope, the echo's
Got
fixed.
it.
I
Got
noticed the echo seems to only come on when we're doing like response videos, so it's got to have something to do with that, like another video in the background or something. But sounds like it's fixed
All
now,
right.
Jeremy. So take your time, brother.
Okay, so if you guys recall, the chimp genome are not sequenced entirely, even as we speak right now, due to budget problems. So their bias and the preposition again uh, made them assume human chimp ancestry, which branched off from a gorilla. Okay, now I recall a quote from an evolutionist from 2 to 3 years ago on this issue. It says something like this. It is not exactly what the person. I don't know if it's a she or he or a she, but he or a she said something like this, which confirmed that the chimp genome is not fully sequenced, because many evolutionists have said that have falsely claimed that the chimp genome is reliable, but it's not, because it has not been fully sequenced. Okay, so just to support the fact it's not yet a fully sequenced, yet they compare it to humans, is really cheating the way I see it. So it says like this, okay, the evolutionist, as I recall, not the exact words. They say that they are hindered by the problem of the human chimp DNA sequence, okay? Meaning the incomplete nature of the chimpanzee genome assemblies, okay? The incomplete nature of the chimpanzee genome assemblies. And now this is their evolutionary proposition I said a while ago. They can make this clear in their assessment, and permit it due to their bias for the what's the English for that, the uncertainties that it has. Okay, hear that? Uncertainties. This evolutionist is willing to admit that we are not certain about the comparison of the human and chimp genomes comparison. Okay, uncertainties. Okay, uncertainties treated as fact due to bias. And when you say you are uncertain in a court of law, your evidential strengths weakens. Okay, so that will be all. Okay, that's
And
it.
one point I wanted to make too, and great points there, Jeremy, is the fact that yeah, we're gonna share a lot of similarity with the chimpanzee, since we also share a lot in anatomy, physiology, and morphology with the chimpanzee, right? They'll point to that hierarchy, and it's like, yeah, it's common sense that we're going to share more with the chimp than we're going to share with the dog or the fish. And to make it clear for the audience, this is what's important about it, is that even some of these very similar genes that you see between the humans and the chimps, what's fascinating is the fact that these similar genes are actually expressed very differently, and those expression profiles bear literally no signs of selection or having evolved, which means, like they said at the beginning and Praise pointed out, these similar genes, although similar, demonstrate a common design, because the function and the expression on those genes are very much different. For example, genes associated with like the brain, for example, or many different metabolic functions show far different expression levels, as I was saying, between the humans and the chimps. You can find different expression levels as well in like the testes, cell signaling, for example, between the humans and chimps. So and what's fascinating too, it's kind of like irreducible complexity, because these genes occur in incredibly elaborate networks, and not only must be precisely coordinated within these networks, but also between organs all over the body. So if an evolutionist points to similarities in genes and says, you know, these were inherited from a common ancestor, just point out the fact that sure, there's similar genes, but guess what? They're expressed extremely differently in the chimp and the human. And that's actually a direct prediction we would make, is that a lot of these similar genes would have different types of functions. So that even that paradigm is falling apart.
Yeah, just because everything has to grow, you should expect that they would have a lot of similarities, but because everything grows, that doesn't mean that they grew from the same common ancestor.
Right. Good point. And let's not forget that we talk about this all the time. And I've never had a good answer for it in a debate. The Y chromosome. When they first sequenced the Y chromosome, they were incredibly shocked at how dissimilar the Y chromosome was to the human Y chromosome. They said this is what we would expect between a human and a chicken. Yet it's, I think the size of the Y chromosome is even more different too. So the so-called 70% dissimilarity is probably even far less than that. And that should be like the Y chromosome is immune to recombination, right? And we know that it mutates faster. But if the chimps and the humans split from a common ancestor, say 6 million years ago, why does that faster mutation rate in the Y chromosome, why do those differences only go back just 4,500 years to Y chromosome Noah? You know what I mean? There's no sign of any chimp human ancestry in the Y chromosome, nor is there in the mitochondrial DNA. They both go back to biblical time frames, and then we've talked about this over and over again. So yeah, you know what? That's all they can point to is similar genes, but those are expressed differently. And all of your major important markers like the Y chromosome, the mitochondrial DNA, they demonstrate a biblical creation model of Adam and Eve. So the genetics is on our side, guys.
Yeah, it certainly looks like we don't have the same fathers that chimps have. That's for sure.
That's for sure, brother. Anything else anybody else wanted to add, or?
Not really.
Well, there are a lot of things to add.
Okay.
There we go.
If you want to continue.
Humans receive only 2 or 3. This is a big deal. Chromosomes each contain thousands of genes. When one chromosome gets deleted, there are very serious consequences. Since chimpanzees and humans are predicted to have only diverged 6 million years ago, there shouldn't be a difference of thousands of genes between them. 6 million years is just not enough time for a significant change like that to happen. So what does this mean? Does the missing chromosome in humans disprove the theory of common descent? Well, no. As it turns out, this missing chromosome is actually a powerful piece of evidence in support of the theory. While completely deleting a chromosome has huge consequences, there's another way that the 24th chromosome, present in chimps and other apes but not in humans, may have gotten lost. Under certain circumstances, 2 chromosomes can become fused without changing how their genes are processed. The location of a gene on a chromosome doesn't change how it's expressed, so sticking 2 chromosomes together is something that could easily happen within 6 million years. So the mystery of the missing chromosome gives us a really elegant way to test the theory of common descent. If Darwin's theory is correct, we would expect...
But okay, so maybe I'm just a layman on this, but this seems to me if there was a fusion, wouldn't they both have the same amount of chromosomes?
No. So what they're saying is and Jeremy, you can explain it too. So the chimp human common ancestor, let's say 6 million years ago, would have had 48 chromosomes. Today we see humans have 46. Chimps and the other great apes have 48, for example. So, why this discrepancy in chromosome number? So they, predicted that maybe there is a fusion, for example. So the chimp line split, boom. Chimp line goes one way, human line goes another way. At first, whether it was for 1 or 2 million years, you can never get a straight answer from the evolutionists, but both lines would have had 48 chromosomes. But at some point in the human line, there would have been this chromosome 2 fusion, and then it would have had to been passed on, they'll say, through genetic drift to every single human on the planet. Therefore, we now have 46, and the chimps and other great apes have 48. So no, we wouldn't expect the same number because the fusion happened after the split in the human line. Does that make sense, Praise?
Yeah, I think so.
Yeah, but wouldn't the information be like wouldn't that make it like a bigger chromosome?
Like the fusion? Well, they'll say that the telomeres at the ends of the chromosomes would have degenerated to because the area is very degenerate. So that's kind of their explanation. But here's the thing I want to point out is that question you made, Praise, actually brings up another point. Is they're looking to the so-called chromosome 2 fusion as one of their better lines of evidence for human ape common ancestry. But even they will admit that this occurred in the human line. Therefore, it's indirect evidence because it's not like it happened before the split, which means even if there was a human chromosome 2 fusion, it could have occurred pre-flood or post-flood around the time of Babel, before the worldwide dispersion, for everybody to get it in time. But the evolutionists would have to explain how they could have, a bottleneck, or how genetic drift could make every single human being on the planet have the chromosome 2 fusion. So, I mean, me and Jeremy are about to get into the discrepancies showing that it probably isn't even a fusion. But it's just funny because even if it is, even if you gave them the benefit of the doubt or agreed to disagree, it's easier for us to explain a fusion in the human line because we have a couple bottlenecks in our model, the flood and Babel, where it could easily be passed on. They have trouble explaining how it could be passed on. I've asked Conspiracy Cats that, and he kind of avoided the question because it's not an easy explanation for the evolutionists. Go ahead, Jeremy. I know you're going to say something, brother.
Yeah, I'll just make it short and in a nutshell. As we know, telomeres prevents fusion. Okay, that is one of the evolutionist problems, and they got rescuing device for that. And those telomeres are, like you said, did you say it? These telomeres are so degenerate in the fusion site itself. Okay, and why are there no? And there's lack of telomere in those sites also. And why are they so degenerate? And don't forget that if you say degenerate, it doesn't mean sorry. If they are degenerate, it means there are not enough telomere sequence as well.
Right.
Right. Okay.
Well,
This helps.
Well,
Yes.
And I'll just play devil's advocate. So, because I've debated, I mean, Erica, I think we had 32 minutes worth of chromosome 2 fusion talk in our last debate. So they'll argue that, okay, since healthy telomeres help to prevent end-to-end fusions, okay, these telomere to telomere fusions, like you were saying, they'll say that both of the two fusing chromosome tips, these telomeres, for example, were severely degraded, with possibly even both of the telomeres degraded to the point of near extinction. So therefore, they're not able to do their job now sufficiently. So bam, you get the telomere to telomere fusion. So how would you respond to that, Jeremy?
Say again. You mean the ends, right?
Well, because the job of telomeres is to prevent fusion. A healthy telomere, that's its job, right? It's the end caps of like, think about the ends of your shoelace, for example, right? Well, over time, what happens, that little plastic piece kind of degrades and it starts flaring out, for example. That's kind of like the telomeres, right? Ultimately, the telomeres shorten over time. So you pointed out that, the job of telomeres is to prevent fusion. And I always ask that too. My first debate with Erica, I said, okay, why were the telomeres involved in a fusion if their job is to prevent fusion? That's usually a lead-in question to see if they know what their next argument is. Because if they don't, well, then boom, the debate stops there. But typically, so a more informed evolutionist will respond with, okay, yeah, you're right, that is their job. So they're proposing that those end caps, those telomeres, were degraded to the point of almost near extinction, which means now they can't do their job because they're not healthy anymore. Therefore, they were able to fuse. And the area is so degenerate now because the telomeres before they fuse were already degenerate. So how would you respond, Jeremy?
A degenerate telomere cannot perform a fusion. Have you heard of this standing for truth?
Well, I've heard that the telomeres prevent the fusion, but if they're degenerate to the point where they can't do their job, now there can be a fusion that takes place.
One point to make would be that without this fusion, evolution would be false.
Right. So, so here's the thing. So that's their answer. So my question typically is, okay, I think that that's weak, but it is theoretically possible. But I would say, why would both of the fusing chromosomes be degraded to exactly the same point, literally yielding a minimized semi-symmetrical fusion site? You know, so it's kind of a stretch, but the fact is the area is degenerate. Oh, go
Yes,
ahead.
I remember. Sorry. I'll let you finish.
Okay, so I was going to say, so yeah, that's what they'll say. They'll say, okay, so the area is degenerate because those telomeres at the time of the fusion would have had to been degenerate anyways for the fusion to even take place, or else there wouldn't have been able to be a telomere to telomere fusion. But so there are some sequences there. They'll point to, I think it's like a 798, and I should pull it up, but I think it's about like a 798 base pair region. So there are some what looks to be like some internally telomeric like repeats. But what's funny is those internally located telomere like repeats that are at that region, they've actually been found to occur throughout the entire genome, from only just a few repeats in length to literally hundreds of repeats long, which and these repeats are not even associated with fusions in these areas. And there's a few papers that suggest that maybe they're involved in like expression, and maybe some of them have multiple functional roles. So yeah, those telomere repeat sequence. So even if you just say, okay, fine, I find that your explanation is weak, that those telomeres were degenerated to the point where, okay, boom, fusion. Okay, now let's look at that degenerate region. Well, those so-called sequences are not are distributed widely over the human genome and not necessarily unique to fusion events. And there's good evidence that there's activity in those regions and possibly involved in expression, for example, like they're binding to things called transcription factors, which then initiate the copying process of a gene into RNA. So that even their rescue device is easily refuted, is my point. So I know I talked a lot there. The floor is yours, Jeremy.
In short, we found out that they are not as degenerate as their explanation is not as accurate as the data that we have found out. We found out that those end caps that you're talking about is not as degenerate as they are claiming. That's the answer, right?
Actually, say that again. Are you saying the telomeric regions are not as degenerate as they're claiming?
Yes.
No, they'll admit that it's degenerate because 798 bases that are seen there, it's an extremely tiny signature for an event. But their explanation is, okay, they had to be. Oh, is there some Jeremy, is that background noise coming from you, brother? It looks like
Gavin.
So it is, Gavin, you got background noise coming from your end, brother.
Look, I'll mute myself, and there's no noise from my side.
No, it's fine. It's fine. It was coming from, and it is good that we spend some considerable time on this fusion event. So we haven't even gotten to the fatal blow. So one major problem. There's multiple major problems that they have. So for one, those internally located clusters of what they say are the leftover telomeric sequences from the fusion, they're a lot more common than people realize. And they're found throughout the genome, evidence that they perform functional roles, for example, expression-related roles, and they're not necessarily indicative of a fusion, for example. So on to the next. The one fatal blow that I typically bring up in discussions, I brought it up to RJ Downer recently. He had no explanation for it, is the fact that there's these ape-specific satellite DNA sequences that are found in ape chromosomes, for example, but these this specific satellite DNA is not found flanking the human chromosome 2. There's a paper, it's the Ventura paper, that goes over this in great detail, because the presence of that chimpanzee and gorilla specific satellite DNA sequence in their genetics, but completely absent in humans, is not only a major problem for the fusion model, it's also a major problem for human evolution to begin with. Now, what's funny is the evolutionists will say this, okay, so why is this ape-specific satellite sequences, why are they absent in man? Okay, because realistically, there should be some of these sequences found flanking the reputed fusion site if we're related to apes, okay? So they'll say, well, you know, it's been maybe 3 or 4 million years since the fusion, so maybe due to mutations or selective pressures, those satellite sequences that were once there at the time of the fusion in the human line, maybe they were lost. Or maybe the sequences were never there, but independently in the chimp lineage, they had some type of de novo origin. So what's funny is either way, they have a rescue device, what's called time did it.
Or there's one more possibility. Maybe we don't have the same common ancestor.
Exactly. And that's the one they don't want to admit. What's fun, because think about it, and you got somebody like ESE, emotionally stunted emoticon, he just can't, he doesn't understand it. Like, why would we expect those to be there, right? Think about it. Even according to their model, okay, 6 million years ago, you got the chimp human common ancestor. Okay, this common ancestor would have had this very distinctive and very large sets of satellite sequences at the ends of their chromosomes. Okay, so now the two lines split. Boom, you got the chimp line going one way, you got the human line going the other. Both lines still have these very large satellite sequences at the ends of their chromosomes. Then all of a sudden, the fusion happens only in the human line. Okay, well, the satellite sequences should still theoretically be there. Now, at some point during that line, they would have been lost because we don't see them. But they won't even question the theory, because guess what? Maybe humans and apes aren't even related, and all it is a rescue device. They have no predictions as to how much of this would have had to been lost in how much time, for example. And here's what I really find funny, and I pointed this out to James Downer. If this ape-specific satellite, these sequences were found flanking our chromosomes and more specifically the reputed fusion site, what would they say? They'd say, oh, here's more evidence for the chromosome 2 fusion. They'd love it. They'd brag about it. They'd talk about it in every debate. But since it's not found, now they have to say, well, why would we expect it to be there? It should have been lost. So do you see how no matter what the observation is, they're just going to force fit it into their stupid little philosophy?
Eventually, when they compare, when they're able to compare everything, they're going to be able to identify a specific human genome, and that the chimp genome is not the same genome. Basically, sorry.
No. At any point, you guys.
Standing for truth, you have explained it very well. The satellite part, I like that. I heard that before from you. But since we are making a comprehensive video here, may I complete the entire argument, the simpler ones for the audience?
Of course, yeah. Chromosome 2 fusion is a major one they use, so this one deserves a lot of time, to be honest with you. So let's be as comprehensive as possible. So go ahead, man. Whatever thoughts you have, let's demolish it.
Things that came to mind. Okay, the gene which say prove the fusion, you know this, does span the fusion site. Okay, I mean this chromosome here and then this the chromosome here, which fuse. There is a same pattern of genes from this side and the other side. So if a gene is unique on the site before, and again I said before the fusion, then why do we see them on the other side of when the fusion happened? Okay, it means it's just one chromosome gene. It's just one chromosome. Okay, because you see similarities and uniqueness between those two fused chromosome. Okay, so it's just one chromosome when you incorporate this evidence, and they have rescuing device, of course. And there are more chromosome samples of this. It's not only chromosome 2. Okay, you can even go look at the straight line version of the chromosome, compared it side by side. There are also not enough telomere, by the way, in the middle of the fusion site, as you know. Like lack of glue to glue two things. Okay, like like for the audience, it is like the fusion site is like a glue. Okay, and there's a lack of glue to glue the two things. That's the way I see it. Agree? Do you agree, standing for truth?
100%. Yep.
Okay. To finish this off quickly, all right, and some don't even look like telomeres, right? Remember that? Okay,
Right.
those telomeres. Yeah, some don't even look like telomeres. So if fusion really happened, and now we're talking about centromeres. Centromeres at the I wish I could share something here. I wish I prepared a slide or something. I didn't really prepare this anything. I was sleeping. Uh,
No
and
problem. And that's what's good.
the central
We don't know
the,
what we're expecting. We just get it. Oh, go ahead, Jeremy.
yeah, if a fusion really happened, now we're talk first we talk about the telomeres, standing for to talk about the more complex stuff, the one that really evolutionists can answer. But what I'm saying here is the things that evolutionists can answer, but they can answer, but it is just a tie. It is not really disputing what I am saying.
Right.
Okay, it is just based on worldview. So if a fusion really happened, the centromere now, sorry for repeating myself, I'm trying to speak English quickly.
Of
I'm
course.
weak at English. The centromere at the upper right side of the the fusion, okay, up the at the upper right, it's really hard without any slide to show. The centromere at the upper right side, at the upper right side. After the fusion line, centromere should still be active, but it became inactive, which goes against fusion because it's a line, right? It's like a pseudogene that it's just an example of the pseudogene. The pseudogene should have been passed, so this centromere should also be passed. Okay, and we do find the lack of centromere in uh, in the line where standing for two. Do you understand what I'm talking about?
I do. I do. You're
Yeah,
doing good. Keep going, brother.
yeah. I really wish I could share a picture here. Okay, okay. So like I said, after the fusion line, centromere should still be active, but it became inactive. So it's like a pseudogene.
Well, the
Okay.
centromere, well, and just so the audience knows, because two active centromeres would be detrimental to the organism. So when the fusion took place, one of the centromeres would have had to be deactivated. Boom. Now we only have one active centromere. So now they need to identify in that reputed fusion site region evidence for a deactivated or cryptic centromere.
Yeah, and what we said is breaks the line, correct?
That's right. Well, they
So
do
it's like
point to an area. They do
the,
point to an area that they say is the cryptic centromere.
yes, so that is their worldview. So like I said, what I'm saying here is the more simple stuff, and they have their worldview, but what you said a while ago, they cannot answer. Okay,
Right.
so it's like a pseudogene, for example, that is supposed to go all the way, okay, But this centromere has kind of like what's the English for it? Has kind of what is the English standard for truth? What you said, the stop it stopped.
A deactivate.
Yeah, it was deactivate. Okay,
Right.
so that's not supposed to happen. Okay, so after the fusion line of the centromere was compromised, so not compromised, but cut or deactivate. Sorry for my English. Cut or deactivated, where they should be active, but it became inactive, which goes against the fusion, right? You agree? It goes against the fusion, right? So, right?
Are you wait? Are you saying that one of the centromeres that are deactivated goes against the fusion?
Yeah, because it breaks the line. Okay, because there are
No.
several centromeres.
No.
It breaks
Because
the line.
if two
After,
chromosomes fuse, if two chromosomes fuse end to end, a telomere to telomere fusion, like they say, there'd be two activated centromeres present, but that would be detrimental to the organism. Therefore, one of the centromeres would have to be deactivated.
Yeah, and there are two centromeres over there.
But one of them would be
one is active and one is deactivated. Remember?
right. Yeah,
Okay.
but that fact wouldn't be inconsistent with the fusion. That's what they would expect.
Really?
Of course, yeah, because two active centromeres is detrimental to an organism. So they say that the second centromere, the cryptic centromere, I say cryptic because that's the one that it's like, where's Waldo? Where's that second centromere if there was a fusion? It would have to be disabled. So they pointed to a region, okay? It's about 170 base pairs long. It's a repeat. And it characterizes primate centromeres. And Tompkins has pointed out that that region that they point to that is supposedly that second centromere, except in a disabled form, is way too small. It's too human to actually be a remnant centromere. And this entire sequence, okay, that they say is the so-called disabled second centromere, this is the fatal blow, okay? It's internal to a functional gene, just like the so-called fusion site is internal to, is a promoter for a functional gene. So that would actually be impossible if the so-called region there was ever a real centromere. So that just completely destroys their idea that it's the remnants of an ancient centromere. And so now what's amazing too, because now we still have those sequences we have to deal with, okay, that region, that 171 base pair repeat, just like the region with the telomeres was highly degenerate, okay, but there's still a region we have to deal with. Well, the telomere-like sequences is not actually the remnants of an ancient fusion. They're found all throughout the genome with strong evidence for function. Now, these alphoid sequences found at the so-called cryptic centromere site are actually a type of sequence commonly found in the human genome, just like the telomere-like sequences, outside centromeres. And they're labeled as higher order repeat. So yeah, this is the fatal blow. And for the audience, if you ever go up against somebody on the fusion, the fact that the cryptic centromere is actually a functional region inside a protein coding gene, okay, encompassing both protein coding and non-coding regions, is strong evidence that it is a key genetic feature to our genetics and not some leftover remnant or some cryptic disabled centromere. So every single one of the observations that should be found, okay, remnant telomeres, a second disabled centromere, they have all been overturned. They've all been refuted. The satellite DNA that's found in chimpanzees and gorillas, but not flanking the reputed fusion site, that absolutely destroys the model. And me and Jeremy haven't even gone over the fact that, well, we did kind of briefly, unless you have something to add to that, Jeremy, but did you want to go over the fact that the reputed site itself is actually overlapped by a functional gene? Did you want to go over that fact?
At the fusion site itself, is that what
The
you are referring?
well,
Over
they point.
the degenerate site is overlapped by a functional element, right?
That's right.
Yeah. I haven't studied chromosome 2 for a long time. It was you who made the better argument that I made. I have last thing to say about this chromosome 2, which came to mind right now. Just one last thing. Um, there was a prediction before that we should find more centromeres in certain areas. Okay, but the prediction failed. It's lacking. Heard of this? Standing for truth. There was a prediction that there should be more centromeres in
There
certain
should be more.
areas.
I've never heard of that. There should be more centromeres where in the gene in that area.
In the yeah, I wish I really have a picture here to show.
Well,
This is the
what is a centromere? It's the center of a chromosome, and the telomeres are the ends of the chromosome, right?
the centromeres are, yes, the center of the chromosomes, and at the center of the chromosome is the telomere, which they say
Okay.
was a fusion site.
Right. The telomeres are the ends of chromosomes. So therefore, when they fuse together, okay, so you picture one shoelace and then another shoelace with those little plastic ends, smash them together. So now you should expect to find those ends now in the middle and not on the ends. So that's what they expect to find, the telomeres in the middle of the site. So the region they point to, as we pointed out, is degenerate. They say, well, we expect it to be degenerate because it had to be degenerate for there to be a fusion. And we say, okay, fine. That's a rescue device. It's kind of a tie at this point. But the fact that that little tiny degenerate region is actually found all throughout the genome and found to be functional in expression, for example, that just shows that these are functional DNA elements and not remnants of ancient fusion. And the centromeres, there should now be two. One activated and one deactivated. So I'm not so. Why would they predict there to be more centromeres? I don't understand that one.
The centromere that you are talking about from is this is. Can you see my hand here? This is the first chromosome, right? And this is the second the chromosome. Okay. There's just a fusion site over here at the middle. So there's a centromere here, and there's the centromere here also. One is active and one is inactive. Okay, the prediction was there should be more centromeres. Okay, in that line. Okay, but it's lacking. Okay, you heard of this?
Are you saying centromere-like sequences, or actually more than two centromeres?
More than one centromere, because this is the last argument. Because this is the last thing I. Sorry about the sound. I think you guys heard it. There. Uh, uh, yeah. Yeah. Where was I? I really want to make this quick so that the others can speak. Yeah. Centromere here inactive and centromere here inactive. Okay, and in the middle is the telomere. Okay,
Well, how many centromeres are in chromosomes? Because I always thought that the centromere is at or near. It doesn't always have to be right in the middle, but
in the middle, yes, in the middle of a chromosome, yes.
Right. So if two chromosomes, if two chromosomes fused, there. So now that chromosome 2 should have two centromeres, one deactivated and one activated. So I don't. So you're saying that they thought there'd be more than just two?
Yeah, That was that was their prediction. You can check Dr. Tompkins' data. I guarantee standing for truth. The prediction was that we should find more. The prediction and again on centromere, there is a prediction that we should find more of it on certain areas, but it's lacking. Okay, I promise you. You recheck Dr. Tompkins' work. Even even Dr. Georgia Purdom agrees. Okay, I think that is my.
I think what you're confusing, Jeremy, is okay. So you have a centromere. Okay, so now if there's a fusion, you'd have 2 centromeres. Therefore, if there should be an area of a deactivated centromere, but what is found is far too small to actually be like sequence-wise, because what they point to is 171 base pair repeat region, and Dr. Tompkins points out that area is far too small and far too human to actually be a remnant centromere. And the fact that entire sequence is internal to a functional gene really tears that apart. But so I think that's what you mean. I think the region that should be a deactivated centromere is too small, is what Tompkins points out, to actually be the remnant of a centromere. Is that what you mean?
Almost. Thank you for clarifying, my friend.
No problem, brother. No problem. That's what I'm here for. One last point on the fusion.
That will be all. That will be all. That will be all. I have nothing left to say. You have provided. Yeah. Okay. That will be all. Good job. Good job, my friend.
Thanks, brother. You too. You too. So I want to point one last part. So the fact that reputed fusion site itself is overlapped by a functional gene means that so-called reputed fusion site is not a fusion site at all. It's a functional DNA element or a functional component of what they call the DDX11L2 gene. And that so-called fusion site lies within, and you've pointed this out before, the first intron of that gene. Have you heard about this argument, Jeremy? So there's been some criticism, though, that says, okay, if that reputed fusion site really is just a functional region, okay, they'll try and downplay the significance of of what Tompkins points out as promoter function and activity in that region. A lot of critics, they'll compare the activity of the non-coding DDX11L2 gene to that of protein coding genes. And they'll have these diagrams up showing expression levels. Conspiracy Cats did it to me. And they'll say that the expression levels are, and the activity in the fusion region, which is non-coding, we know that, is extremely limited compared to the protein coding genes. So they're trying to say that it's not functional. Have you heard that before?
So you're talking about the, let's say this is the fusion. Okay, there's introns and exons on both sides, right? That's what you're talking about, correct?
Right.
All right. So yeah, I agree with everything you said.
So they'll say that it's not okay. So here's what Tompkins points out. He points out that these critics don't realize that long non-coding RNA genes, they cannot be properly compared to protein coding genes. And here's why. It's because long non-coding RNA genes are expressed at much lower levels. Therefore, it's not good method to compare those and try and say, hey, listen, this area has low expression levels, therefore it's not, it's not, there's no activity or function there. So that biological importance can actually be measured simply by, which the critics do, comparing them to protein coding genes. And I like the way Dr. Tompkins points out, he says, and I'll just quote it, he says, this is an important detail that ill-informed critics don't realize, because he's compiled data that includes includes epigenetic marks, transcription factor binding marks, transcription start site data, expression levels, co-expression data with other genes. So Dr. Tompkins points out that all of the genomic data in combination, when you look at it as a whole, which is contradictory to what the critics are doing because they're so desperate. They're so desperate to hang on to this being an actual fusion event. When you look at it all in combination, they all corroborate on one clear fact that actually demonstrates that this alleged fusion site really is a second promoter in that DDX11L2 gene. So boom, we've destroyed the telomere-like sequences that should be there. We've destroyed the fact that there should be a cryptic centromere. We've destroyed the fact that there should be some ape-specific satellite DNA flanking the reputed fusion site. And we've shown conclusively just how functional this region is. So they're really hanging on to very little here, Jeremy.
Don't forget the Synteny thing, which is on our side. You know about that, right? The
Which one, brother?
thing.
Which one? Synteny.
The Synteny thing.
Yeah,
The
you know what?
chromosome, the
Right.
Synteny thing.
Hey, can you cover that one? Just so we can, like you said, make it as comprehensive as possible.
Yeah. It is inconsistent. I really. Do you remember the exact data about this standing for truth? I'm kind of lost here.
Oh yeah, no, don't worry, brother. Brother, you can leave it to me.
Yeah,
man. This issue.
you do it. I'm kind of lost regarding this, but I remember there's one more. It's the Synteny thing, okay?
Right.
Because I think we want to make this comprehensive. So I recall there's the Synteny thing also, and I think that's the last one.
Yeah, and it's the fact that there's no homology where there should be around the fusion site. There's lack of homology is what you're getting.
Yeah.
Thank
you for reminding me.
No problem.
Thank you for reminding me.
No problem. I just think it's funny because they'll say this is in praise. Just think about this. They'll say this is a fusion site. Boom. Telomere to telomere fusion. Smack them together. Well, guess what? You don't achieve incredibly important genes and incredible function by just boom slamming together two chromosomes. So, but most importantly, even if there was a chromosome 2 fusion, they will admit that it occurred in the human line. Therefore, it's indirect evidence. And think about this. Let's just say there was a fusion, okay? Well, the fact that there's no ape-specific satellite sequences found flanking the region, okay, that's, and Dr. Sanford brilliantly pointed this out. He said, fine, fusions in general are evidence for genetic entropy because fusions, believe it or not, are actually mistakes. You can get fusions in cancer cells, mistakes in the crossing over process, for example. I mean, these are evidence and demonstration of the fall in genetic entropy. So even if there was a fusion event, since there's no ape-specific satellite sequence DNA there anyways, okay, it's evidence for a human-only fusion event unrelated to chimps and humans, and we can explain it better based on either post-Babel or post-flood, since we have two bottlenecks. So for the creationists out there, don't be scared of this piece of evidence because there's a lot of data, as Jeremy and I covered, that refute a fusion. But even if there was, don't be scared because it could easily be explained according to our model. Did you have anything to add there, Jeremy?
I think we covered everything, so we can move on. Good job.
Thanks, brother. Good job to you too. So I know we're being comprehensive, but I learned that from Jeremy. When it comes to their best evidence, Jeremy, what do you say? You got to be comprehensive, right?
Yes, just like the gravitational time dilation and the nested hierarchies we were talking about yesterday, the other day. And this video also talks about pseudogenes, as I recall. So nested hierarchies, in a different point of view, we will have to discuss when we reach that point later.
Right.
Yeah.
In the video.
They always talk about reutilization of
Any
the
question?
genome. they always talk about the repurposing of genomes. For example, they say that while copy was made, or in this example of the fusion, they say that it was repurposed, but they don't really have any evidence of that ever happening, do they?
Sorry, repurpose in English is what again?
Like they'll say you make a copy of the genome, you have an extra copy of the same information, and then they'll say, well, that information is going to be repurposed for some other use, but that's not really ever been observed. They're just making it up, really.
So you're talking about gene duplication here, right?
Yeah, and in this case too, where they put the two chromosomes together, and then now you have a functional gene in between the two chromosomes, and they would have to say that was a repurposing of the genetic information.
That's a great point, Jungle. Yeah, that's, it's good. I'm glad you brought that up, because that's where their philosophy comes into play, right? That's what they always look to, the repurposing of certain sequences. Yeah, so they'll say, I've had them say, I think Erica too, like, so what if the region's functional? Is it impossible for that region to become functional at some point? Just like the ERVs, the ALUs, the retrotransposons, the pseudogenes, you point out the fact that these are incredibly functional to healthy life processes in the cell, for example, and in the embryo and stuff. And they'll say, well, as you said, Jungle Jargon, these functions, or these genetic elements have been repurposed to be functional, or these functions have been co-opted. But once again, no evidence for that. That's their philosophy.
Yeah, very important.
Great job. I think we're on. How long left is this video? Praise. Oh yeah, I can see it. So it's still very long. So I think we better move on if we really want to destroy everything.
and we may just have to do parts 1 and 2, because you're right. When you get us in here being so it sucks that they had to use chromosome 2 fusion as their first, because that always requires a lot of time. So maybe
that's not their best evidence.
No,
that's not their best evidence.
No, I agree. It makes no sense that, because
actually, their best evidence are distant starlight and nested hierarchies. Their second evidence are this chromosome fusion and dating methods. So best evidence, starlight and nested hierarchies. Second best evidences are this chromosome 2, and the dating method. So that's all.
Actually, that's a good point. We're going to do probably a dating method one in the next few days. And Jungle Jargon, you're good at that too, brother. So we'd love you to be there. We've done distant starlight recently. We've done nested hierarchies the other day. We're doing this one, and hopefully we'll do dating methods soon. So Praise, if you're still there, brother, you can click play at any time. You there? Praise,
no,
you there?
I'm not. I just got back. My bad.
Praise, did we lose you at the chromosome 2 fusion?
Yo.
I mean, Praise thought that.
Can you hear me?
Praise's argument.
Next slide.
Well, I mean, shouldn't we all have 48 chromosomes? But so Praise, I hope you learned something there, because never use that argument in a debate, brother.
Yeah. There's a lot of information poured out.
So you learned something there?
Yeah.
Good.
I learned, let me see, this is so much that was before, so I have to go back and learn. Go back and hear again.
At least hopefully you learned why they'll say humans have 46 and
because they have to say that.
Dummies.
Yeah, I think we have to move on. I think we have to
move on. Human chromosome that looks a lot like 2 chimpanzee chromosomes fused together. But if no such chromosome is found, then the theory would be seriously undermined. The answer comes in the form of human chromosome number 2. What we find is that chromosome 2 is very different from all the other human chromosomes. In fact, it looks exactly like 2 chimpanzee chromosomes glued together.
And we already refuted all this. So yeah, just keep it playing. Well, we're not going to rehash that all again. So just keep it playing.
Let's strike up another conversation on this, guys.
I know. But hey, the
Located
second
near
we
the
heard chromosome 2 fusion, we just destroyed it. Actually, you know what? Don't fast forward. Just let it play through so the audience will see the lines of evidence they use. For example, the telomeric regions. You'll see them cover all that, and you'll see how we demolish that. So let it play through, please, and then we'll get to the
middle of
next one.
every chromosome is a special region called a centromere. This unique structure is an attachment point for something called the mitotic spindle. It allows duplicated chromosomes to be pulled apart during cell division. Centromeres are made mostly of repetitive DNA sequences, and they're very easy to recognize. Normal chromosomes have and need just one centromere structure. Chromosomes also have DNA structures called telomeres at their ends. Telomeres are important because they protect the ends of chromosomes from falling apart or from fusing with other chromosomes. Like centromeres, telomeres are easy to identify. They have recognizable structures with special repeating sequences of DNA. All normal chromosomes have a single centromere, usually near the center, and 2 telomeres, one at each end. But chromosome 2 is different. It has not one but 2 centromeres, one that is functional and one that has mutated a bit and is now non-functional. It also has not 2 but 4 telomere sequences. Strange as this seems, the story gets even more interesting. These extra telomeres and centromeres are arranged exactly how you'd expect them to be if 2 chromosomes were stuck together head to head. They're arranged as follows: telomere, centromere, telomere, telomere, centromere, telomere. As you can see from this diagram, this is just what a combination of 2 chromosomes ought to look like. Also, it turns out that chromosome 2 genes and genetic material found on 2 chimpanzee chromosomes, 2A and 2B. So, the unique structure of human chromosome 2 completely fulfills what we would expect if humans and chimps do in fact share a common ancestor. Its odd structure is best explained by the theory of common descent. In other words, the most reasonable way to explain why chromosome 2 looks so strange is that a human-like ancestor long ago developed a mutation that fused 2 primate chromosomes together. Sometimes it's argued that chromosome 2 has these extra sequences because it was specifically designed that way to carry out some function that we've not yet discovered. But the vast majority of scientists agree that this is an ad hoc argument. According to our current knowledge of biology, there is absolutely no reason that a chromosome should have extra centromeres and telomeres. The most reasonable explanation is that chimps and humans share a common ancestor. Another way to test the theory of common descent is to look at special pieces of DNA called pseudogenes, or false genes. Basically,
Pause that real quick.
Pseudogenes are
So I hope everybody's seen that she pretty much, well, we wouldn't expect there to be function there. Well, guess what? Function's been found. She said we shouldn't expect any extra centromeres, telomere-like sequences. Well, we just spent almost 45 minutes destroying the fact that they're rebuking their claim that those areas are found, and the very, very small regions that are found are all found throughout the genome, not associated with any type of fusion event. So do you see how Biologos supposed one of their best evidence, each point that they made, we systematically demolished, and they didn't even touch on anything regarding the function. They just went right over it saying, well, we wouldn't expect there to be any function, or it's just post hoc, ad hoc. I mean, do you realize how much function and activity really is there? So yeah, I hope everybody noticed that real quick before we move on.
Yes, I noticed.
Okay, keep
Can
going
we-
there. Yeah, it looks like we're on.
Play the video. Continue playing the video.
Play there, Praise. Praise, wake up. Click play.
genes that are no longer able to make functional proteins as most genes do. A small percentage of pseudogenes do have some sort of function. Some of them help to regulate the expression of other genes, how often those genes are translated into proteins and when. But usually, only a small portion of the pseudogene actually plays a role in that regulatory function. The rest of it doesn't seem to matter to the organism. In any case, whether they have a role in gene regulation or no apparent function at all pseudogenes are a permanent part of the genome, and thus they get passed down from generation to generation, just like normal genes do. The genetic mutations that produce pseudogenes occur at random, and can affect nearly any part of a gene. There are
Yes.
hundreds or even thousands of different individual pieces called nucleotides.
Regulation is a huge thing. It's not a small thing.
Yeah.
Go
Thank
ahead.
you for stopping. I just have a little thing to say about that. What the girl said. Evolutionists predicted that the DNA is littered with pseudogenes, right? Yes, true, but slowly becoming not true. My English is really today. Many of those pseudogenes are no longer pseudo because they are functional after all. Evolutionary rescuing device. Okay, as we know, we talk about it. Is the turning on and of those pseudogenes. When you say turn on, when you say turn off, then that means it becomes a pseudogene. Then when you just say turn on, then it is functional. Okay, so it has been turned on and off so many times in evolutionary history, depending upon the environment. Okay, they have their long time scale, so this is their benefit. Okay, that's why Kenham and Answers in Genesis, top 3 research, because they got priority researches. One of their top 3 research is to disprove the evolutionary long time scale. Because, yeah, Kenham said that many times. You have to disprove the long time scale. One of the 3 things I just trying to let the audience know that one of the top 3 things that Answers in Genesis is pushing is to disprove the long time scale. Not chromosome 2, okay, not the others, but the long time scale. Although I do agree partially with that, I do not completely agree. But that's what he's pushing, the long time scale. Now, because the long time scale is one of the primary ways evolution is possible. Even if evolution is possible, if the planet is only 6,000 years, okay, then evolution even if evolution is possible, but the planet is only 6,000 years old, then there's no time for evolution to happen, even if it's possible. Okay, I hope my English is doing well. Okay, so take away the long time scale, evolution is dead. Dead. That's one route we can disprove pseudogenes also, because pseudogenes do form what's called a nested hierarchy. So when I say YE, it's Young Earth Creationists should break that hierarchy. Okay, one is through function.
Jeremy, real quick, nested hierarchy. I've never heard of that. Can you explain?
What? You never heard of that?
I've never heard of nested. No, I've never nested hierarchy. First time I've ever heard of it. What is that?
You're joking, right?
I'm joking. We just spent 3 hours.
Raw man in the house. What's going on, Raw man?
Raw Matt, my man with the plan. Hey, how's it going, brother? What's living the dream?
Yeah, just getting ready for tomorrow. A little crazy day.
Hello, Matt. Hello there, Matt.
Hey,
What do you got tomorrow?
I just have last things. Hello, Matt.
Yeah, Jeremy, I was just making a joke. Take your time, man. So this is especially the pseudogenes is your expertise. Take as much time as you need.
Yeah, so Young Earth Creationists should break that hierarchy. One is through function, and that is from Dr. Nathaniel Jeanson. Like I said, yeah, and another is to prove that the function of one pseudogene in a hierarchy, or sorry, there's an echo again. There's a problem with the sound. Can you guys hear?
Yeah.
Yeah, but
there seems to be an echo problem again. Yeah, I think there seems to be an echo, Chris.
I'm not sure where. Like, I don't hear it on my end, so I don't know.
Any guys here? Yeah,
Yeah,
never again.
I
yeah.
can hear the echo. Yeah, it's
Yeah. You can hear it sounding for two. There's a problem,
Praise.
right?
Praise, did you do anything in that moment differently?
What?
I think I was.
Because I wanna explain it clearly.
I was trying to.
I wanna
What did you
explain
do?
it.
What did you do differently in that moment, Praise? We'll figure it out right now.
I was stargazing.
No, but
Nothing.
really, what did you do differently in that moment? Because it's coming from your end. So
I hit the Streamlabs. Maybe I'm supposed to use Streamlabs, or I don't know. So we'll see.
it's definitely something that just try and think what you do differently. Okay, so Jeremy, talk again.
I forgot where I was.
Yeah, you see how it's on now, Praise. So you should be able to narrow it down. So whatever you were just in, Praise, every time you go to that seems to create an echo. So
Yeah, so I remember where I was. So YE creationists or Young Earth Creationists should break that hierarchy. One is true function, as you know, standing for truth, from Dr. Nathaniel Jeanson, and you do pretty well. We do pretty well. And like I said a while ago, another is to prove that the function of one pseudogene is in a hierarchy or a hierarchy. Okay, let me clarify. Function, okay, like I said a while ago, and another is to prove that the function of one pseudogene in a hierarchy or a hierarchy which extends from one animal kind to another is different. Okay, you prove that this hierarchy is functional, okay, in one family, and you also prove that this function within this family is unique from the function hierarchy of another family. Okay, you know about this for truth, right? I told you this a long time ago. All right. So um, hierarchy from this, let's say this insect here, there's a hierarchy, and we have to prove they are functional. Okay, we're slowly beginning to successfully do that. Okay, but these functions, okay, that we found in this hierarchy, also let's say its hierarchy extends beyond another insect, another type of insect. Okay, let's say this is one insect, this is one insect, but they both form a hierarchy which looks like common descent. Now what we have to do here, okay, to make it easy to understand, is we have to prove that this hierarchy here is not only functional, but its function has to be different from the other family, the other insect type, the other insect family. Okay, so more research has to be done. Okay, but we are slowly beginning to successfully prove. I'm really trying to do my best in my English and speaking fast so that the rest can speak. So the function, that is the first step, and we are headed there. Okay, but the uniqueness of the function, yes, we are also there, but it's a long way to go. I cannot say that we have completely or even closely. I can say that we have not successfully completely debunked their nested hierarchy. I have to be fair to evolutionists too. Okay, I have to be fair. So nested hierarchy is hard to disprove. I told you this, standing for truth. Okay, so that is why it is one of the their top two evidence: distant starlight and nested hierarchy. Okay, so that's it. Okay, so you prove that there is function of one pseudogene in a hierarchy of one animal kind, and the hierarchy of the other animal kind that links the two different animal or insect kind, which extends from one animal kind to another. Their functions should be different, and not only they should be functional, but their functions should also be different. Okay, that's the best way to disprove it, and we are slowly doing that, but it will take a long time before this is completely disproved. Okay, so I have to be honest. We'll have to be patient. Maybe we will never see the time when this is completely disproved, because the DNA really is so complex, and we have studied only less than I think less than 3% of the DNA DNA language. Okay, so again, function and unique function are the ways to remove the nested hierarchy. That's it. That's it. Unless you want me to go even further. Any questions?
Nope, we'll get going here.
Right.
Yeah, and I'm not sure. Are you guys getting kind of tired? Like it's kind of late, but if you guys want to do more, I'm up for it. If it's up, this is up to you guys.
So what? I'm gonna say more.
Sure, yeah.
Because I remember that the next part of this, because Standing for Truth showed me the video in the morning, but I forgot some of it. But I recall pseudogene is not only the argument that the video was talking about. It is also talking about the pseudogene that is shared between ape, human, and chimp. And I think the video that we are showing here has not reached that point yet. But this is a critical issue. So if you want me to proceed with this in advance, we can do it. But if not, you could just play the video. So it's up to you guys. Because I've read Dr. Venomous book, so I have a more or less idea how to refute it.
Yeah.
Yeah. So
yeah, I'm fine. Keep going. Yeah, Praise. You can keep going a bit more.
Similarly,
yeah.
there are
So I'll just
thousands of different
explain
ways that
later
those
when we
pieces
reach that
or
point.
nucleotides can be switched around or deleted to convert a normal gene into a pseudogene. Since there are so many different ways that a pseudogene can become mutated, it is incredibly unlikely that the same exact pseudogene would become mutated in the same way, in the same specific place, more than one time. If a specific pseudogene with certain mutations is found in the same location in two or more species, we can conclude that most likely those pseudogenes came from the same place. In other words, that the two species descended from a common ancestor. So what does the evidence say?
Here?
Do we ever find that two or more species do advance the field of genetics?
Yeah, I'm sorry. I just want to make it clear for the audience. Just quick. Remember, Standing for Truth, we talk about the evolution of the English language, right? Remember that. The idea of this is kind of like the evolution of the English language, which this girl is talking about. That in one generation, let's say for example, the word let's say praise. Yeah, praise. Okay, the word praise in one generation it is spelled differently. Let's say it is R. There is no letter R, for example. Okay, but it is still praise. Okay, in one generation, then another generation, the meaning is still praise, but another letter has been changed. Okay, or added, something like that. Remember that, Standing for Truth? If you want to help me out here. Then another generation, another letter has been changed. Another generation, another letter has been changed or deleted. Anything. Okay. So that is the point of the girl over there. That in every generation, a certain gene would this part would change, that part would change, this part would change, this part would change, but it will still remain the same gene, same gene, and to the point that because they have their millions of time scale, this gene would become completely something else. That is how their evolution, the evolutionary explanation, Okay, so I'm really sorry. I'm having a hard time speaking fast with my weak English. So I hope everybody understands. So let's say we have a sentence here. Praise is a good man. Okay. Praise. Letter R was removed. Okay, but it's still praise is a good man, but letter R is removed in this generation, in the 14th century, for example. Okay, and then in the 15th century, then praise is a good man. And in that generation, the word man is still, the meaning is still man, but another N is added. Okay, or maybe letter A has been removed. Okay, okay, so am I explaining it Do you guys understand? I'm really having a trouble, explaining it very well in English and in a very quick way. Am I making any sense?
Yeah,
Standing for truth.
changes, the changes made affect the future generations.
Yes, it is
Of
like the evolution of the English language. You guys remember that? Know that, right? It is called descent with modification. Okay, in the 14th century, the English language. I think you guys know that in the 14th century, the 15th, 16th, 17th century, the English language has kind of like changed, right?
it's no
Yeah,
longer
you know that, right?
understandable. The old
Yeah.
English, you can't even understand it. I can't.
Yeah, exactly. You are right. Whoever you are, I'm sorry. I I'm confused
Hello,
with
hello,
with
Jeremy. I'm Jungle.
yeah, so forgive me. So that is the concept of evolution. Okay. So I just want the audience to, since this is a comprehensive video, I just want the audience to fully understand the evolutionary side and what they're talking about. It's called descent with modification. The English language is the descent with modification. The modification is, for example, like I said, the removal of the word "r" in the word praise, in the word praise, and the addition of man, another letter N for man. Okay, so it's the same concept with the evolution of the English language. Okay, pretend that each word is the word that is classified in the uh, i- in the groups of nucleotides. Okay, because there are words, letters, chapters, and an encyclopedia of the DNA. Okay, so picture every word as the word as the word and letters. Okay, and those letters and words do change, and that's called descent with modification, and that is how the English language also evolved throughout the centuries. Also descent with modification. Okay, so that's it. Sorry for I'm doing my best to really make it quick and with my weak English. So forgive me. So sorry. Any questions, or we should proceed? Sorry for taking time. I just want the audience. I really want the audience to understand. Okay, because
No, I think you're doing a great job, Jeremy.
yeah. If you want to add something, because standing for truth, you know that we both talked about this before. So it's up to you guys. I'll stop now. Sorry.
No, I think you did an exceptional job on that topic. So it praise. If you want to move on, you can.
Place. In other words, that the two species descended from a common ancestor. So what does the evidence say? Do we ever find that two or more species do have the same pseudogene? The answer to that question is a resounding yes. Chimpanzees and humans, for example, have a great many identical pseudogenes with the same exact crippling mutations. Also, they're in the same exact spots in the genome.
Actually, praise. If you want
The
to
beta
pause it
type
real quick,
globin gene cluster.
I'll make one small point. Even
Yeah.
though I may even link the nested hierarchy video Jeremy and I did the other day, because we go into so much detail, Jeremy, as you remember. But even her saying, crippling mutations, a lot of what she's saying in terms of genetic mistakes, quote unquote, just like all evolutionists, they assume that all the DNA differences in our genome are the result of mutation. While we reject that idea and we propose a created heterozygosity and front-loaded DNA diversity. So, Jeremy, as you pointed out, we know so little of the DNA language. It's like if I were to go to Russia, and we explained this the other day, as a proofreader, and I know nothing about Russian language, I'm not going to be able to point to a book and edit it and say, hey, this is a mistake. That's not a mistake. That's what they're doing in the genetics right now. They're pointing to what they believe oftentimes are genetic mistakes because for a number of reasons, maybe they have some signatures of broken protein coding genes. But guess what? The more we study the genetic language, the more we realize, oh man, these are not genetic mistakes. These are functional DNA elements A lot of these pseudogenes, when they're damaged, they lead to disease. And Matt talks about this oftentimes. If you're damaging something that's functional, it will oftentimes result in disease. That's a good way to find out if certain gene sequences or certain DNA elements are functional. Oftentimes, you can just mutate them or look at natural mutations and see if there's. Go ahead, Jungle.
There's one thing. There's only one thing that's evolving, and that's their claims of evolution that are evolving. No actual evolution in the genome, but it's their claims that are evolving. As they just make claims going as they go, and then they just correct the claims as they go. And so the only thing that's really evolving is their claims.
Right. And I would say because their claims are getting weaker and weaker, their claims are devolving.
Exactly.
Rescue device after rescue device. But so let's just say, okay, these genetic mistakes, we see that they're being turned over more and more. And Jeremy's right. We know so little of the genetic language. And they've done so few knockout tests. That's the gold standard of genome function in regards to testing. So there is still a lot of work to be done to identify functional roles for a lot of the pseudogenes. But what we do know about the fact that these pseudogenes in bulk are inherited from generation to generation. So we at least know that they are being transcribed, they're being passed on from generation to generation. Natural selection is not weeding them out. Therefore, if they're just wasteless junk, useless evolutionary leftovers, then natural selection should have gotten rid of these pseudogenes or all this supposed junk DNA long ago, because it's wasteful of resources for the cell. But my point is, so if you see these pseudogenes and we know that they're functional and they are necessary to sustain healthy life processes in the cell, okay, if we share some of these functional DNA elements with the chimps, with the other great apes, with the old world monkeys, all the way down to dogs, fish, and we can see some hierarchical patterns, well, that makes sense. Since we both expect some level of hierarchies, well, then we're going to expect to share more functional genes, functional DNA elements like the ERVs with the chimp than we would with an old world monkey or a dog or a fish. That's why we say, no, these patterns reflect function. And therefore, that's why we look to function and we predict more function. So I just wanted to point that out. I tried to condense 2 hours worth of information into just a quick few-minute explanation. So did you have anything to add to that, Jeremy?
Yeah, actually, since the girl already brought up the human chimp pseudogene. I think somewhere the next few minutes or wherever in the video, she will also talk about, she will extend the human chimp pseudogene into the gorilla and the gibbon and so on. So she is going to provide stronger evidence afterwards. And I've read it in Dr. Venema's book. Okay, human chimp. Okay, for now. Okay, as we know, let's just forget about the human chimp overall genetic difference. Chimps have let's say 95%. We are only let's say 80% similar with chimps, right? Standing for it, is it 80% or 85 or 90? What is the exact number?
It's about 88% when you. It's 98%. They'll say when you.
It's 95%. They say 90. But
Yeah.
I want to ask you about ours. What do we say? I forgot what ours is. Sorry. Is it 80%? If you really
If you consider the gaps, the copy number variations, the insertions, deletions, and the size differences, for example, it's about 88% is what we.
88%.
88%.
I have to recheck that because I made the calculation before, and I heard even Dr. Georgia Purdom said that it's 80%. But anyway, that's not really the point. So the girl in the video and what you said about the human chimp, I think the next part that she will be talking about somewhere along the line is it gets more complicated because she will link the human chimp to the gorilla and the gibbon and the other great apes. Okay, so this is in Dr. Venom's book. So this is so let us not use those overall similarity between humans and chimps because we can really disprove that, right? Standing with truth, but by those gaps and so on. Okay, so let me try to make it harder for the evolutionists here by providing specific numbers and using specific evidence in which I don't know if I've read it in evolutionary literature. It's been a long time, really. I don't know if I read it in the evolutionary literature and came up with this with some ideas, or I read it in ICR or CMI, but not in Answers in Genesis. Anyway, sorry for saying those things. I just wanna
Well,
just ask any questions, okay? If you do not understand, because this is really technical. Even me, I'm still studying it even as we speak. Okay,
I
I'm
just
still not.
I wanted to point
Yeah,
out.
sorry.
No, that's okay.
Yeah.
About the DNA similarity. So, yes, the researchers, they disregarded major portions of both chimp and human genomes because those portions they were looking at raised some difficult questions. For example, they'll look at like an area in the genome and then another area in the genome that seems to be duplicated from that from another area in the genome. So their difficult question would be, should we count that or shouldn't we? So what they did is they excluded, instead of answering those difficult questions, they just decided to exclude a lot of those large, well, I think all of those large mismatched sections, which is actually over 1 billion letters. So if you do a letter by letter comparison on the remaining, I think it's about 2.2 billion letters. Both the human and chimp genomes are about 3 billion letters, are both the same. So they look at just 2.2 billion because they're excluding all the other large mismatched sections. Then, within that 2 billion letters, 2 to 2.2, they find between 95 to 98% similarity. Okay, fine. But that's only if we ignore the other 18% of the genome. But they'll justify it as, okay, humans, chimps, they split 6 million years ago. Therefore, we're going to expect changes since then, right? Gene duplications, deletions, insertions. So they're trying to, because obviously, even if common descent was true, we shouldn't misrepresent their model. Of course, there's going to be some change within 6 million years, right? So for one, they're assuming common descent. So they justify excluding those regions. For us, we don't assume common descent. Therefore, the 88% similarity is fine with us. But I always point out that instead of arguing all day about the percent similarity, I just say 88%, 80%, 95%. It doesn't matter because what is true is that there still is a hierarchy. So regardless of the percent similarity, chimps and humans are still a lot more similar than humans and the old world monkeys or humans and dogs. So regardless of what the percent is, there still is a hierarchy. So I always say it's the hierarchy that we need to deal with. And that's why we deal so heavily with it. So I just wanted to point that out, Jeremy.
Yeah. Like I said, we have to again for the audience. We have to not only to break the hierarchy. Like I said, we have to not only prove that the hierarchy that extends between one family of animals to another, but we also have to prove that they are functional on both sides, on both families. But we also have to prove that their functions are unique, are different.
Right.
Okay, that's the only way. Okay, so now going to the pseudogene ape human chimp gibbon thing, in which the girl will later talk about because she already started the human chimp thing. I remember it is somewhere next to that. And Standing for Truth already started the human chimp. Now I'm gonna extend it. I'm gonna complete it by incorporating the other great apes. Okay, now first of all, there are really so many examples I can give, but I could give some more familiar examples for the audience, in which I believe some of you guys here already heard. Okay, pseudogenes that are shared between ape, human, chimp, gibbon, um, gorilla, and so on. That is why they suggest invoke common ancestry. Okay, let me give one example of how these pseudogenes can be broken apart. Okay, I'll represent the evolutionary side properly here first. Okay, for example, chimp and us have the same DNA sequence for the beta globin. Sorry, beta globin pseudogene, right? Correct? Am I right? Is it beta? Correct, right? Chimps and us have the same DNA sequence for the beta globin pseudogene, but this is pseudogene looks like functional actually. If you look at it, okay. If you look at it, okay. When you look at how it how it forgot the English for that. Sorry. Okay, but it looks like functional. Okay, but we do share that pseudogene. Okay, if it's not really a pseudogene, that is only a degenerate one, and a degenerate DNA element is not really a pseudogene. The way I see it, it still works. Okay, a car. Okay, like I said in the previous video, a car that is very old can still be used. Okay, so it is degraded just like a gene degraded, and they use the word pseudogene for degraded DNA elements, and that is misleading. Okay, so okay, sorry for that. Let me continue. So this beta globin pseudogene that is shared between humans and chimps looks functional. Okay, why? Because it only looks degraded. It is not completely a pseudogene. Okay, okay, so it varies, okay, this pseudogene looks like functional actually. Okay, but it varies. Sorry, I'm really doing my best to speed this up with my weak English. But this pseudogene looks like functional. Okay, it looks like functional. It looks degraded. So it's not a pseudogene, like I said. So that's why I explained the car thing a while ago. So so this pseudogene looks like functional actually. Sorry for repeating. Okay, but it varies that but it varies for it also varies that it sorry for stammering. It uh, Hindi, kasi pwede lang kung uwi kasi lang. Hindi, wala uwi kaila. Sorry, I was talking to my mother. Hindi ako makikitsong nila sige. We're speaking Chinese here. Sorry. Pseudogene looks like this beta globin pseudogene that chimps and us are have similarity of. They say it's a pseudogene, but the way I see it, it is not. It's just a degraded gene. Okay, but they are misleading, so they say it is a pseudogene. Okay, so this pseudogene looks like functional actually, but it varies. It varies that this allows it. Okay, but it varies that this allows it from becoming protein. Okay, I think some of you understand this, standing for truth. But it varies that this allows it this allows it from becoming a protein. So they think it's a broken gene. That's one reason why they say it's a pseudogene. Okay, okay. But when you look at it in another worldview, it is just degraded. It is not really a junk. It is just a degraded gene. Okay, but
are you saying, so for the audience sake, Jeremy, are you saying, so say you get a car off the assembly line at the moment, so once you start driving it, everything for the most part is working near perfectly. The air conditioner is working nicely, the heater, the brakes. So over time, let's say, I don't know, let's say you've had that car for 6 years and suddenly let's just say the air conditioner and the heater is not working as well because it's essentially degraded since its initial design. So just because it's degraded doesn't mean that it's not working at all. Is that what you're
Yeah,
saying? It's okay.
yeah. Thank you for clarifying for the audience. My English and my speed of talking made it more complicated. So thank you, Standing for Truth, for making it.
You're doing good, brother. No, I got it. I just wanted to clarify.
I hope the audience also. Yeah, thank you. I also want the audience to get it. So just interrupt me when I get more complex and more deep into this. Okay, or raw mat anybody there. Okay, so this I really hate to say pseudogene, but because this is not a pseudogene, it's very misleading. Okay, so the errors. Right, sorry. All right, I'll just use the evolutionary term here. So the so-called error of this beta globin pseudogene are in the same DNA sequence for both humans and all the great apes. Okay, in the same area and the same sequence. Okay, so first impression? They share it. They pass it on. Okay, so it looks like evolution, right? Okay, so they think that the error, this beta pseudogene, this for me is only degraded, came from the same ancestor. But in fact, these so-called errors are so very similar. But the fact that these so-called errors are so very similar do suggest that design function. It's a design function after all. Okay. Am I saying it right? So the errors are in the same DNA sequence for both humans and ape. Sorry, I already said it. Sorry, I already said that. So they think that suck. I really shit. I already sorry for cursing. I already said that. But the fact these so-called errors are so very similar, okay, do suggest that there's designed functional elements after all. Some past claimed broken genes. Some of the past claimed. How do I say this properly in English? Some in the past. Okay, some past some past pseudogenes that are claimed to be broken genes have been discovered to actually play important functions like regulatory roles. Okay, I will not. I already explained it in the other video the other day in detail. So I'll just use one example here. So this may prove to be the case with the betas. Sorry. This may also prove to be the case with the beta globin pseudogene. First, it could be transcribed into it could also be transcribed into RNA. Okay, and wait, RNA. RNA transcripts itself might be useful, okay, it relates to this beta pseudogene. Even if it is not made into a protein, like I said, because it prevents from becoming a protein. That is why they think it is a broken gene. Um, this RNA transcript itself might be useful even if it is not made into a protein. Sorry, just clarify if I'm making a mistake or getting more complex. Secondly, the RNA transcripts that I was talking about, it's not transcripts in other very similar similar pseudogenes have been found to regulate the transcription of nearby genes. Okay, so again, so is it pseudo or not? You decide, okay? I say no. This is not pseudo, okay? An example is when I can give one example out of many examples. Babies. An example is mammalian. Hang on. Sorry. An example is mammalian babies. Okay, when they are inside the womb, any mammal. human, or others, animals. I really hate to label ourselves as mammals because we are specially created. So we are not animals. We are not mammals. For me, okay, to me, an example is when mammal babies are inside the womb. Okay, unique when they're inside the womb. Um, Unique types of hemoglobin ensure their tissues get oxygen. Okay, correct me if I'm wrong, guys. Please, interrupt if you guys do not understand or see some mistakes. Okay, because I do not completely remember the data here. Okay, it's really been a long time since I studied this, but I'm a little rusty. Okay, so mammal babies are inside the womb, unique types of or different types of hemoglobin ensure their tissues get oxygen, and when the baby is born, it switches to adult hemoglobin. Okay, it switches to adult hemoglobin. Okay, so the regulation when and which the regulation when and which these globin genes of them are turned off and on. Okay, my grammar is having a problem again. So where was I? The baby, after the baby get oxygen, and when the baby is born, it switches to adult hemoglobin. Okay, so the regulation when and which globin genes of them are turned off and on. Okay, turned on, so there is function. Turned on, there is function. Okay, see, here we go, function again. All right, turned on, so there is function. Now I'll give some numbers I recall from 6 years ago. Okay, I hope it's really messy. I have an archive of those specific numbers, but I'm just so the numbers may not be accurate. It's a more or less. Okay, so forgive me, correct me if you know the numbers. Okay, I will no longer use function as a tool here. I'll provide numbers, okay, to make it harder for evolutionists. Okay, now and to make sure this reaches them, those A's and these numbers, okay, so that they can challenge me, and I'll challenge them in a debate also. Okay, now a large fraction of most of the pseudogenes differ hugely from their homologous genes. Okay, again, a large portion or fraction or whatever the English is of most of those pseudogenes differ hugely from their homologous genes when it comes to their nested hierarchies. Okay, standing for truth, you're following. Okay, okay, again, a large fraction of those pseudogenes, okay, those nested hierarchies, differ hugely from the homologous genes that they are claiming are nested hierarchies. Okay, differ hugely from their homologous genes when it comes to their nested hierarchies. Okay, so when they say nested hierarchies, it is not really as as scary as it as as many people think who understands the nested hierarchy idea. Okay, so
Yeah, like,
okay, an example, sets of let's say
yeah, I don't want to jump in right now, but I'm starting to
yes,
like fade away.
you were
I'm
saying.
starting to fade away and getting tired. I would love to finish like this tomorrow if you guys want to come on. I mean later today if you guys want to do that.
Okay, okay, I got a small point to make. Maybe if there is such a thing as nested hierarchies, then it would
Yes,
it would reasonably be true that if you took a zygote of a human and put the DNA of a chimp inside of a human zygote, that it would be compatible, but it's not compatible.
Right, that's a good point. That would be like in vitro fertilization, and to be, I think they've tried that before, way back in the day, which is weird just to even think about. But
Right.
in
But
vitro should be able to, especially if chimps are our most recent common ancestor. You'd think
well,
they'd be able to.
yeah, and part of the point is they still don't know how the proteins are folded, and at least one human protein is folded differently,
Yeah.
and that's determined. That information is coming from somewhere other than the DNA in the humans, and it has to be a human genome has to be in a human zygote to be compatible to function.
Telephone.
So
Yeah, I think what you said relates to the homologous genes I was talking about that. And yes, Mr. Jungle Jargon. Telephone. Sorry. Uh, okay. Homologous genes. Okay. Quiet. Uh, okay. When it comes to okay, an example. Yeah, I remember where I was. An example. Let's say a set of six. An exact. Okay, I'll speak a little louder because someone is using the phone over there. An example: sets of let's say 60 plus primate pseudogenes. Okay, so we're talking about several primates now, not just human and chimps. Okay, several all of the ape family. Sets of 60 plus primate pseudogenes. Okay, I forgot which blocks they are in. A DNA sequence, their DNA sequence indicates that the parts of the pseudogene sequence are the same almost to their homologous sites, but not higher than but not higher than the random chance levels. Okay, so we here see random chance evolution is compromised within this human ape, gibbon, and so on. The entire ape family. Okay, it's 50% for two unrelated strands of DNA. Okay, less than one third of the based on Dr. Dennis Venema, and I could turn it around. Less than one third of the sequences, which are 85% similar to their homologous sites, and only little unknown section or fraction of the same reach only 90%. Now now the lower. I'm really doing my best to make this fast. Now the lower level of similarity mean greater or different interpretations. Okay, so take note of that. Okay, so there is a different worldview around here. So there is a tie between us and them. Okay, and I believe Dr. Venema knows this. That is why he is a Christian, a theistic or evolutionary creationist, a Christian. So he is more honest. Okay, but he is also partially biased. Okay. Okay, so take note. This is a different. So this is a different interpretation of the hierarchy of the pseudogenes of hierarchical so-called pseudogene samples. So the origin of the timing, the origin and the timing of the accumulated gene of the accumulated pseudogene or gene differences. Okay, now the logical conclusion of this. Okay, the logical conclusion. This means that shared mistakes arguments is severely weakened and sometimes invalid. Okay, so this is one way we could debunk the hierarchy other than the function argument standing for truth, and that Dr. Nathaniel Jason is pushing. We use specific numbers over here. Okay, okay. Again, so this means that the shared mistakes argument